Anybody need the handouts from the last class? No. Okay, so lecture 5. So review session after Thursday lecture for those that want it. We'll take a break somewhere around 11:50. Assignment 2 will be due this Thursday. You've got a Word document up on Blackboard if you want to use that. And you've got the hard copy I gave you last Thursday. So last time we talked about proteins, specifically the building block of proteins. We have the generic amino acid. It's got a common backbone. The nitrogen, carbon, carbon running through every single amino acid. And what is different about each of those is the R group sticking off of that middle carbon. And what that that R group does is give the amino acid its character. Does the amino acid like water or not like water? Can it be reactive? What's there, what's not? Its chemistry will determine how it acts within a protein within that chain of amino acids. Specifically for folding, we join those amino acids together for a dehydration synthesis reaction. We reverse that by doing what? We add water, and that process is called what? Anybody remember? Water breaking hydrolysis. So dehydration, we pull that water out. We connect those two amino acids together to form a peptide bond between the carbon and the adjacent nitrogen. We add water back, we can break that bond. The proteins can flip around, can turn around along that backbone. And how they turn is largely dependent on whether the R group likes water or not. So we've got the top group up here that generally doesn't like water, hydrophobic. It turns away from water. It tends to crowd together. And then we have the other ones that seem to like water. They will turn toward water. So that molecule, that string, will fold up in such a way that the hydrophobic is on the inside and the hydrophilic is on the outside, because every cell is full of water. So the example we gave in class, if we've got the R groups, the blues that like water, we've got the R groups in, I guess it looks sort of tannish, maybe pinkish, don't like water, they are going to turn away. So that's the protein or the peptide that's been formed. As it empties out of the ribosome, it's going to start to flip and rotate. Yes, sir.
Does the R stand for ribosome, or?
R stands for generic, those 20 different ones we talked about. Okay. The R groups are here. So you can have an R group that's as simple as one little hydrogen, or any of these other options.
Now, is there any specific name for them, or just the R group?
When we talk about them collectively, they're called the R group. Okay. When they're attached to a specific amino acid, that R group gives that amino acid its character and therefore its name. Okay. So if you've got just a glycine, a hydrogen, that becomes glycine, the amino acid glycine. Whereas phenylalanine, we got this ring sticking off, that becomes phenylalanine. Okay. So each of these 20 amino acids has a different R group sticking off. So we can have the different R groups bending in different directions. Those pink ones are going to try to get away from water. They're going to try to hide from the solution that surrounds that protein. The R groups will say, hey, I like being around water. They're going to go out to meet it. So that straight line starts to bend. And eventually we get a collection of R groups that are all huddled together trying to exclude water. And then the blue R groups, the ones that like water, the amino acids that like water, tend to be forced to the outside, tend to cover the shape of that protein. And it's the interaction with water that determines what the final shape of any given protein is. Every protein starts out as a straight line. And then those amino acids, the R groups on those amino acids react in water. We've got our amino acid ribbon or sequence that makes the protein. As it reacts with water, we start to get sections that form sheets, sections that form helixes. So different sections are meant to be spiral. So the inside of those spirals, we probably have R groups that are hydrophobic. The outside R groups are amino acids that like water. These sheets, which are pretty much flat, there's a lot of glycines in there. They tend to be hydrophilic. They like water because there's no place for a hydrophobic group to hide. We get those individual building blocks, we develop a more complex structure that is the final shape of a protein. It's done folding, everything is where it wants to be, everything is happy with its relationship with water. It's near water, it's not near water, however that do there. And then we have proteins, a single protein will sometimes group with other proteins to form the next level of structure. So you'll see that in muscle proteins. You'll see that in our blood. Hemoglobin is 4 different globs stuck together, 4 different protein chains. You're going to see that quaternary structure a lot of different places. So the first thing we call with protein structure is the primary structure, just the sequence of amino acids, that chain as it forms. Once we have that chain coming out of the ribosome, it starts to fold up as it responds or reacts to the water, which is the solution it's in. We start to get the hydrogen bonding that forms the alpha helix or the pleated sheets. That is the secondary structure. Certain sections of a molecule are meant to form sheets. Certain sections of the molecule are meant to form coils. Hydrogen bonding is the key. The stickiness of one hydrogen on one side to another amino acid somewhere kind of holds things together. So we've got the initial folds, initial sections forming different structures. Then we have those structures interacting with each other to form the final shape of that protein, the tertiary structure. The overall final shape of a protein, and they generally fall into 2 categories. Are they globular, little round balls, or are they fibrous? Are they long sheets? Are they things that are strung out, flat, or stringy? So that's just one chain of protein, how it folds on itself, how it comes together, gives it that tertiary shape. Then if we have multiple proteins coming together, we have that quaternary shape where 2 or more individual protein chains come together and interact. They form some sort of structural unit that can be or can do a variety of different functions depending on what the needs are for that function of that protein. So you can see how one determines the other. Which amino acids we have present? Do they like water? Do they not like water? In the order they are is the first level, and then that determines each interaction after that. So this is an example of the peptide chain coming out of the ribosome. We have those individual dots there are individual amino acids. The color codes are meant to show you where things end up at the end. So we have that primary structure reacting with water. It's going to fold, it's going to twist, and we're going to have our folded shape at the end, the final shape. This protein going toward more of a globular protein. So we have those 4 proteins formed. So you have a string of amino acids. We don't really see the individual amino acids in this diagram. We're assuming we've got thousands and thousands. So as it gets in the water, those chains start twisting, shaking, until it finds a stable relationship with the water. So we're looking at 3,000 nanoseconds. That's about 3 milliseconds. That's 1,000th of a second. that all this happens. So automatically, its reaction with water, its reaction with bodily fluids, creates our final shape, whatever that may be. So a slight change in amino acids is going to change how that protein eventually folds up. If I take one amino acid out that, say, was hydrophobic, and I replace it with a hydrophilic, that's going to change that section of the molecule. So a slight change in amino acid sequence can affect or influence the ability of that protein to carry out its function. The shape of the protein determines how well that protein does its job. And by changing amino acids in locations, we can have different success in those functions. So we've got hydrogen bonds that lend stability to the secondary structure. The hydrogens on one part of the molecule, protein molecule, will interact with other ones and give us some stability. So we can see that across here. You've got your individual hydrogens, oxygens interacting just like they did in water. They're going to stick together. They're going to interact to provide some stability to that final shape. So when we have that carboxyl end, we have oxygen on that end. We have the middle carbon with the different R groups hanging off it. And the amino end, that's got hydrogens. We can have interactions between that oxygen and the hydrogens in the amino group. They're not bonds, but they kind of stick together. They kind of stabilize each other. So those hydrogen bonds, the reason tape tends to be sticky, one of the reasons, hydrogen bonding, helps stabilize that secondary structure, those coils and those sheets. So as I've alluded to, shape is very important in the world of protein. Whatever the shape is will determine ultimately what it can and can't do. So once it's folded up, we have a fairly stable shape that happens very, very quickly in a fraction of a second. We have its function. So we have a genetically governed condition called sickle cell anemia. The difference between those with sickle cell anemia, those that don't have that condition, is the hemoglobin molecule, the subunits that make it up. We take out one amino acid and we put in a different one. Point mutation. That change in one amino acid affects how that protein folds up, and it affects how it can do its job, or what we might see morphologically. So standard red blood cells are going to have that double concave shape, like a donut where we didn't quite finish the hole. The change of that one amino acid in that thousands of amino acid proteins is going to change the shape of the cell. And that's going to influence its ability to carry oxygen, which is going to be a negative. It's going to influence the ability to fight malaria. They seem to do better fighting malaria. So there are positives and negatives associated with mutations. We see how they all work. So the character and nature of protein is determined by the sequence of amino acids. Those amino acids, each different of the 20 amino acids, has a different R group, different character, a different reaction with water. So ultimately, the R groups, or what's hanging off, are going to determine what that protein does or can do. One of the things you'll see out in the literature, protein is the synonym polypeptide. So a number of peptide bonds strung in a row makes up a protein, or we can call it a polypeptide. Everybody with me so far? Make sense? So we say primary structure. That sequence or chain is going to determine how the secondary structure happens, which will determine how the tertiary structure happens. Primary structure, what we're really talking about is the individual amino acids or the individual R groups. So getting that individual amino acid in the right spot is going to make sure that protein folds the way it's supposed to. So in this diagram, primary leads to secondary, leads to tertiary. One determines the other. So DNA needs to be conserved so we get those proteins made the correct way every time. If we are not consistent in our copies of DNA, we would not be here. So the ability to replicate that DNA without mistakes, we'll talk about in a little bit. But that DNA determines what the proteins are. When we have proteins, we have 7 different classes, and they're largely based on their shape, what they can and can't do. We have structural proteins. These tend to be those fibrous proteins, hair, tendons, ligaments. So something that is a building block or a 3D structure that holding things in place, or making something that's helpful at some point. 2, we're looking at contractile proteins, the things we specifically have in the muscle. So they all work together. Same net effect. They have different structures. Some are branched, some are not. But one protein contracting over another allows things to shorten, and the ability to shorten things is very helpful when we're working with our muscles. We'll have storage proteins, proteins that are meant to save amino acids for a later function. So a chicken will create ovoalbumin, globular structure protein in our egg whites that's meant to provide amino acids to the growing chick embryo. If you think about it for a second, we have a very little embryo when that egg is laid. Everything that we need to grow a chicken must be inside that egg, or whatever other creature we're going to do. Is that an amphibian? Is that chicken? Is that a dinosaur? Everybody's finding. It's all got to be there. So you have proteins that are meant to hold that amino acids until that embryo needs them. Transport proteins, things that pick up things and move them around. So hemoglobin is a classic example. It picks up oxygen and moves it deep into the body where it's needed. You'll also have transport proteins for copper and iron, which do not work well being floating out in the blood free. You'll have transport proteins for fats, because fats are not soluble in the blood. So you've got some sort of bus that moves these things around. You've got a class of proteins that are called defensive proteins. These proteins usually aren't pure protein. There are sugars involved, carbohydrates involved. These antibodies are meant to recognize and attack foreign things to the body. We'll have hormones, FSH, LH, thyroid stimulating hormone, all sequences of amino acids, usually a lot smaller, hundreds versus thousands of amino acids. If we think about the defensive proteins, those molecules are huge relative to the body. And the last group we'll talk about is enzymes. We have things that are going to put things together, take things apart, transport things around the cell, start and end processes. They are the machinery of the body, and they are made of protein. So enzymes start processes, end processes, help processes accelerate. They are catalysts to get things done in the body. Enzymes, probably one of the more important roles of proteins, but who's to say that any of those other protein functions are less important? It'd be great to have enzymes, but I think I'd have trouble not having tendons and ligaments, bones, things like that. So all chemical reactions in mammals are regulated by enzymes. So the speed at which they occur, what actually gets formed, what actually gets done is dependent on the enzymes that are present at any given time. And it's really kind of amazing when you think about all the reactions going through your body that have to happen minute to minute to keep you going, to keep you happy, healthy, and a part of the society. Any questions on proteins? Everybody understand the structure and how they form and what the building blocks are? So we'll come back to them again and again. We'll keep running into those things over and over again. So why don't we pull out your letters and see how we're doing with hypotonic, hypertonic, and isotonic. The red blood cell is placed in a hypotonic solution. Water follows solute, so the cell will fill up with water until it bursts. Nothing will happen. The red blood cell will empty of water and shrivel up. So if we got a red blood cell, where's the high solute concentration relative to a hypotonic solution? Hypotonic, where's the high solute concentration? In the cell, where's the high water concentration? Outside the cell, can the solvent move, solute move?
No.
No. Generally, no. We're simplifying. Can the water? Water will move from its high to a low. That gives us answer A as we move through. So it's going to move from inside the cell down its concentration gradient from outside to inside. Hypertonic. What's going to happen there? So where's the high water concentration? Inside the cell. Water can move. It's going to go down its concentration gradient. It's going to leave the cell, which gives us a cell that shrivels up, or answer C. This is starting to become second nature. Which of the following is true? Starch is a polymer of glucoses from plants and digestible by mammals. Starch is a polymer of glucoses from animals and can't be digested by animals. Starch is a polymer of fructoses from animals and digestible by animals, or D. Starch is a polymer of glucose from animals, digestible by animals. So what do we know about starch? Polymer of glucoses. Where is it from? Plants. And is it digestible by animals? Yes, so that gives us answer A. So if we were to put cellulose in there, how would that change our answer?
Not digestible.
Not digestible, so we move down. Well, we don't have that answer, but it would have to be indigestible to make that the correct answer. So next monomer for discussion is nucleic acids. These are what make up our DNA and our RNA. We're going up in complexity. Glucose, relatively simple to amino acids. Nucleic acids become more complicated still. So nucleic acids, we string those together to get DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. And they go together a little bit differently than what we've seen so far, but still have that dehydration, hydrolysis reactions to link them together. Nucleic acids, generally a bigger molecule. It's got all of the big four in it. It's got oxygen, hydrogen, carbon, and nitrogen. So nucleic acids are the genetic material governing inheritance, primarily through the control of photosynthesis. That material, that information is coded in our nucleic acids. There was a movie somewhere that said computers are basically two things, two codes. Everything is a 0 or a 1. Switch is open or the switch is closed. With our nucleic acids, we're moving from 2 to 4. 4 different, well, actually 5, but 4 effectively. So the two polymers that are done here, main interest, are DNA and RNA. So DNA has rings that lose that oxygen. Ribonucleic acid has sugars with those oxygen present. Deoxyribonucleic acid for DNA. Ribonucleic acid or RNA. So the monomer that makes up DNA and RNA has 3 parts. Each of those molecules that are the monomer, the nucleic acid, have 3 parts. So those 3 parts are a phosphate group. We have a sugar. That's what the difference between RNA and DNA is, whether we have the deoxyribose or the ribose sugars. And the third part is the nitrogenous base. So we have these 3 pieces stuck together. And we're thinking about DNA, we're looking at an L-shaped form. Show you, it's on that handout I gave you. We'll show you that picture. So we have the phosphate group in yellow, with the phosphorus in the middle there, surrounded by oxygens. We have the sugar here, so we've got a 1, 2, 3, 4, 5 carbon sugar. And then we have our nitrogenous base attached to that in the green. So lots of N's for nitrogen. So nitrogen, nitrogen, nitrogen. Lots of different nitrogens. How many we have there depends on how many rings we're dealing with, which brings us to our next point, that just like our amino acids, there are fixed parts and there are variable parts. With our amino acids, we had the R groups. There were 20 different ones of those. The variable part, the nitrogenous base, there's only 5 of those. More or less effectively 4 different ones that we can have. So the coding system that makes up you, me, our cows, any sort of animal on this planet is based on the combination or the rearranging of 4 different nitrogenous bases. How we put them in order depends on whether we're going to be a slug or a cow, or a plant for that matter. So looking at this, you have an individual nucleotide. The colors match, so yellow is yellow, that phosphate group. We've got the blue is blue, our sugar group. And we're kind of forming an L here. Our nucleotide comes, nitrogenous base comes off at a right angle from the sugar. And we'll have 4 different DNA, 4 different options. So DNA, our nitrogenous bases or nucleotides. The differences in those nitrogenous bases are going to lead to adenine, thymine, cytosine, and guanine. Those are our 4 different nitrogenous bases, 4 different nucleic acids. With our RNA, we make one slight change. We go from thymine to uracil. The nitrogenous base is the same. We've got a slightly different sugar, but we're not going to worry too much about that in this class. So adenine, cytosine, guanine. remain constant as we move from DNA to RNA. As we switch from one to the other, the thymine becomes a uracil. The standard convention with these is we identify them by their first letter. So A stands for adenine, T stands for thymine, U for uracil, C for cytosine, G for guanine. So the difference is the variable part here, and then whether we've got that oxygen there or not. So when we're doing our nucleic acids, there's a certain few different rules that effectively make sure that the DNA gets copied correctly. So the nitrogen bases or nitrogenous bases show distinct pairing preferences. Adenine always pairs with thymine. A always goes with a T. Cytosine is always paired with a guanine. So if you look at the diagram you have there, again, the color coding remains constant. We've got the yellow is the phosphorus, the blue is the sugar, and then we've got the pairs reaching across. So depending on which side we have, what we're starting with, we have the appropriate pair on the other side. So T with A with T, C with G, G with C. Now, because this is meant to be a spiral, these rungs on this twisted ladder look like they're different lengths, but that's just the point of making it a two-dimensional appearance of a three-dimensional diagram. All those rungs on this twisted ladder are exactly the same length. And that's one of the reasons that the DNA replication is conserved. So if we take a ladder, envision DNA as a ladder, where the sides of that ladder are the repeating unit, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar. That blue-yellow repeating pattern on the side. And then we have the that right angle coming off the sugar, where the nitrogenous bases pair up in the middle to form the rungs on that ladder. I assume that twist has to do with structural integrity, helping hold things together, but you'd have to ask God for that answer definitively. So we take that ladder, twist it, all the rungs are the same length, they're paired bases, and then the sides are the phosphorus and sugars. So we've twisted that helix, we've got the phosphorus and sugar, repeated pattern going up the side. So that is not where the genetic information is. The genetic information is in the rungs of our DNA. So if we take a look at this diagram, you should have that one in your handouts today. The coding is a little bit different from a color standpoint. But if we're looking at this DNA, we've untwisted it, unhelixed it, flattened it out. We see on the sides here, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, forming the side of that ladder. We've got the same on both sides. Then off the sugars, we've got our nitrogenous bases that determine which nucleic acid we have. So phosphorus sugar base, that's one nucleotide. Phosphorus sugar base, nucleotide. Phosphorus sugar base, nucleotide. So just looking at that, what do you see about the interactions between A and T and G and C? And what do you see that's common about each of those rungs? Just physically looking at that, what do you see? We said all those rungs had to be the same distance. So what do you notice about all those rungs coming between those two sugars? You got a sugar on this side and a sugar on that side. What makes them all the same length? You see that? So we've got this space in the middle. These dots are meant to represent hydrogen bonding between an O and an H. So that's the space between your G and your C. That's the space between your A and your T. Going across, how many rings do you have? 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. That rung there is made up of 3 rings. If we put 2 thiamines together, or thiamine and a cysteine per rung, what do we get? 2. That's too short. If we take a guanine, put it with an adenine, we get 4. That's too long. So all of the pairings that we have form 3 rings to make those rungs the same distance across or the same length. What up there indicates why A always bonds with T and why G always bonds with C. Adenine with thiamine, guanine with cysteine. Why is those pairings conserved? A and T, T and A, you've got 1, 2 hydrogen bonds. 1, 2 hydrogen bonds. Guanine and cytosine, 1, 2, 3 hydrogen bonds. 1, 2, 3 hydrogen bonds. So the pairing is about the connections that are made. Adenine and thiamine make 2. So they always go together. Guanine and cytosine always make 3. So they always go together. So restricting it to length or 3 rings, and then matching the number of connections that can be made to the other side of the helix, everything stays constant. If I take out this guanine right here, get rid of it, the only thing that can go back there and make the rung the same length, make the proper connections, is that guanine. I can't bring in a thiamine. We got one, we're short one ring and we got the wrong number of connections. Adenine, if I bring that in, it's the right length, but not the right number of connections. So those two simple rules, every rung has got to be 3 rings. And then I got to match up whether there's 2 or 3. Helps make sure that those pairings are constant, that our DNA is replicated over and over and over and over again without mistake. Or at least it's very hard to make mistakes. Everybody see that? That makes sense? The simplicity, if you think about it, is beautiful. There aren't no complex rules, just 2 simple ones. Then make sure our DNA, if we take apart, we split the helix. This half goes this way, this half goes this way. When we reconstruct on the opposite side, we get exactly the same 2 daughter strands of DNA that were exactly the same as the mother strand. One of the questions I asked up front, does everybody know the connection between DNA and protein? And you were kind of shaky on that. So why don't we take our break right there and we'll come back and we'll answer what is the question between DNA and protein. We'll restart at 6 of. Okay, so let's get back to work. Connection between DNA and protein. We have 4 options in our DNA. We got 20 options in our amino acids. So how do we get from one to another? So we have to do a 2-step process. We can look at DNA as kind of the library where everything is. We make a photocopy of that with our RNA. And then we use that photocopy to build our protein. So DNA goes to RNA, goes to protein. So it's a 2-step process. We go through transcription and translation. The DNA to RNA step is called transcription. We are transcribing. We are making a copy. And the language is nucleic acid to nucleic acid. So we're speaking the same language as we move from one to another. We're making copies using the same language. When we go from RNA to protein, we're translating. We're changing the vocabulary, the letters, the alphabet that we're using. We're going from a nucleic acid language to an amino acid language. So we got to figure out, okay, when we say this in nucleic acid, that means this in amino acid. And how that's done is we take 3 nucleic acids in a row, which is called a codon. And that codon tells us which amino acid we need to use. So that's how we translate. So this is how we convert back and forth, and you have this, you should have this in your handouts. This is how we translate back and forth. Well, at least in this direction. We have a codon. It's got a first position, a second position, and a third position. So this diagram shows us all the possible combinations that occur in RNA. So we can talk about the first position, so uracil, cytosine, adenine, guanine. So all the codons that are in this section, their first position is guanine, first position. All the codons in this section begin with a C, or a cytosine. Everybody see how that works? We start here and then we rotate to the top and we determine what's in the second position. So we're dealing with columns now. So this column, all these codons have uracil in the second position. This column, cytosine in the second position. This column, adenine. This column, guanine in that second position. Then we go over and we talk about the third position and things get a little bit more complicated. We're going back to rows. So this row, we've got uracil in the third position. This row, we've got cytosine in the third position. So what you're doing here with all the codons, 1, 2, 3, 4 columns, is we've got all possible 3 codon codings, so positions. These are all the possibilities that can occur. Now we've got, what is it? There's 64 different combinations. If we do 4, 4, and 4. But there's only 20 amino acids. And what we see in our codons is that more often than not, the first 2 letters get things done. The third can be anything. So if we look at an amino acid here, SER stands for serine. In the first position, we've got uracil. Second position, cytosine, and then it doesn't matter what's in the third position. All of those will code for a serine. So that's if we have these codons present, serine is the amino acid that appears. And that happens quite a lot. Proline's got 4 codons that work for it, threonine, alanine. But we have some other ones here, phenylalanine versus leucine. That third codon becomes important. What's in that third position? If it's a uracil or a cytosine, it becomes phenylalanine. If it's an adenine or guanine, it becomes a leucine. So we have all 20 options here, some redundancy. We also have 3 that act as stop codons that tell the ribosome that's making these proteins to stop. Anytime the UAA, UAG, or UGA shows up, the ribosome stops making that protein. Is everybody following the logic of this? That how a 3 nucleotide codon is matched with a specific amino acid. So that's how we translate. We get from one to the other. So we'll come back to this diagram. We're going to make a very small protein here to understand the process of transcription and translation. When we're doing those processes, we got to make sure that it's transcription and translation versus just replication of the DNA. The DNA, you've got 4 pairs in there that are always the same. When we transcribe and translate, the differentiation here is we drop the T and bring in a uracil. So replication, that's a different process than the transcription and translation. And we're going to focus on making a protein, which requires transcription and translation. So let's start with a simple protein. conservation of pairings, making sure that that rung is the same distance across the 3 rings. And then you've got the 2 paired with 2 and 3 paired with 3. So that's going to make all these pairings happen over and over and over again. So translation, we're going to go from DNA to RNA to protein, or more specifically, DNA to RNA to amino acids. Everybody got that wants it? So we've got 5 codons here from the DNA. So the first thing we need to do is translate or transcribe to the messenger RNA. We're making a copy. We're going to take that copy out, we're going to feed it into the ribosome, and that's going to make our protein. So what does that RNA look like? So what is this? T pairs with A pairs with U, C pairs with G. What do we got here? U. Does everybody follow that logic? So we're just bringing in the corresponding pairs going across. Now that messenger RNA is formed. We're going to use the tRNA to bring in the correct amino acid. So which tRNA do we bring in? So for AUG, what do we bring in? Looking at our table. Methionine, MET. Does everybody see that? You do the A is your first row. Then you go up to the top, you pick your G. So that should narrow it down to a column. And then you've got your last letters of G. So we've got a row first. Then the row intersection, we got the box. And then we've got the last one over on the right-hand side gives us our G. Does everybody see that codes for methionine? So AUG, probably should have moved that closer. So first letter is an A, so that means it's got to be in here somewhere. Second letter is a U, that means it's got to be in this box. Last letter is a G, so that takes us, AUG is methionine. Everybody see that? Lauren, you finding that? Making sense now? Yeah. First time you do it, so this table's a little confusing. But if you practice with it, yes.
Sorry, I didn't mean to. But we have to do that for every single code, like you consider a codon.
What I'm asking you to do is for these 5.
Yeah.
So can we go through and see that UUU codes for phenylalanine? That UUA codes for leucine. That GGC codes for glycine. And then our UAG is our stop codon. We've completed the protein. Now, normally this goes on for thousands in a row, but I don't have that much room on the board. Everybody see how that works? So we've gone from DNA to RNA to our protein. Very short little protein, but it is a protein. we get from transcription, translation. Any questions on that? Have you done this before? Some of you have done this maybe in high school or somewhere in biology. Some of you have not. So let's try it again. So we've got our protein up top there, TAC, TTC, CCG, GTG, ACT. What is our messenger RNA going to look like for that one? Pairings continue up, doing our transcription. A pairs with T pairs with A pairs with U, C pairs with G, G pairs with C. So that's our messenger RNA. That's what's going to go in the ribosome, and that's where the translation is going to occur. And I came up with methionine, lysine, glycine, histidine, and then a stop. Everybody following that? We need another example, or you got it down? Is that a yes for another example?
No, that's a yes,
We
right?
got it down. So what happens when we have a mutation in our DNA? So using that same thing, we can have two types of mutations. One's called a point mutation, where we go through and replace one of our G's, one of our nucleic acids, with a different one. Something happens in the replication of DNA where we make a mistake. We hope that doesn't happen too often, but we replaced an adenine with a G. Guanine, what does that do? So if we look at protein 1, I don't think it made a difference. So the same protein. So depending on where the mutation is, sometimes it'll have an effect, sometimes it won't. In this case, we code for the same thing. What if I go through and replace that adenine with a T? We take that phenylalanine, we bring in a tyrosine, and now that molecule is going to fold up a little bit different. The R group on tyrosine, different than phenylalanine. It's going to react a little bit different. Might still get the job done, might not. But it's going to be different. We translate something different. Probably the most disruptive ones are called phase shift mutations, where we add in a nucleic acid or we take one out. In this case, we're taking one out. So we're dropping one of the adenosines, we're shifting things over, and that's going to mean that every codon after that point has the potential to be different. I'm shifting everything down one or shifting everything up one. In this case, we're going to get a phenylalanine again by luck, but we got a premature stop, so that protein is never going to be made. And I have a friend who has a child, Fox G1, where he can't make certain proteins due to something like this. So that makes things a little different. And he's got some challenges there, as well as his child has some challenges, but they seem like they're reasonably happy. So still things can work. If you get too many of these mutations, too many things not doing what they're supposed to, you can see how the system falls apart. And what we're looking at in genomics is what nucleic acids are where. So you can see that changing a nucleic acid may or may not have an effect. So we saw a protein there where it didn't change anything. And other times you change that nucleic acid, you're going to change the protein. So genomics is all about looking at which nucleic acids are where. So we're thinking about the history of genetics and genetic information. We look to 4 different ways of looking at things. We have the pedigree of an animal. We look at the animal's ancestors and try to interpret what the genetics are for that animal. So based on her ancestors, what will this cow do? We're trying to guess at what the protein is. We might look at performance. What is the animal doing itself? Given that performance, we can infer about the protein or infer about the DNA that she has based on what she is doing. We can also look at the progeny. And this is what we do with our bulls historically. How much milk is in this bull? Well, he's not going to express that with his own performance. He's not going to lactate, but his progeny will. We look at their performance and then we interpret how much milk or what the genetics are of that animal. In all these cases, we're inferring what the DNA might be. Based on probabilities, based on likelihoods, we're guessing what that DNA might be. The new is a direct look at the DNA. What is there, and then what does that mean? So we look at all the positions in a DNA. Where are places where things are different, and where are places that things are not different? So what might you expect to see as differences across cows in our codons? There's probably going to be a lot of variability in that 3rd position. That really has no effect, right? So there might be a variety of animals that have different codons in that 3rd position, different nucleic acids in that 3rd position, but it really doesn't affect the performance of the animals. But there may be some spots in the 2nd and 1st places where we have a different nucleic acid, and that might change the protein slightly, make that animal a little more efficient, a little less efficient. So we look at the DNA, figure out what nucleic acids are where, then we monitor the animal's performance. So animals that have an adenine in that 5,256th position seem to produce more milk. Or those that have a thiamine in the 4 millionth position seem to taller. And that's what we're doing. We'll pick that up on Thursday, so.